Thank you, everyone, for having me today. Thank you for organizing the event. It's very lovely to be here today. So yes, as Paul explained in the first, well, as I was preparing, I'm currently a developer in Bratislava Message Lab. I'm also a PhD student. And today I'm going to present a couple of tools that we've developed. And by we, I mean this person right here. So Ross Marcello is the person that developed most of the tools I'm going to present today. Now he's gone. He's actually a real developer now. He finished his PhD and now he's working in industry. So I took over most of the tools related to neuroscience that he's worked on during his PhD. And I'm going to show them to you today. I'm going to explain a bit what they do. And I'm going to do, so in the first part of this course, seminar, lecture, however you want to call it, I'm going to do an overview of all the tools that we've developed, how they work. And then in the second half, I'm going to do a small tutorial. Well, I'll show you what we can actually do with them. And I'm here on behalf of Bratislava Message, who's my supervisor. So the title of today's, of what I'm going to present today is called Tools for Contextualizing Human Brain Maps, because really that's what the tools I'm going to present today are about. It's about, and the first question you might ask is what do I mean by contextualizing brain maps? Well, the idea is that when we do some kind of scientific research in neuroimaging, oftentimes we'll end up with a brain map at the end of our analysis that gives us intensity levels for some kind of measure. Like, for example, here I'm kind of showing just for an example, the big brain, the layer 4 thickness from Conrad was those work in 2020. And then when we have a map like that, this is just an example, we might want to ask ourselves whether we want to contextualize it basically with respect to other results from other people, from other groups that they've obtained in the past. So we might want to ask, for example, in this case, whether some particular region in that brain map that we have is enriched with gene expression, neurotransmitter receptors, metabolic ideas, cell types, neurophysiological associations, and a bunch of different things. And this might be easy, this is a quite simple question. So we might ask, for example, layer 4, is it the case that brain regions that take layer 4, so for example, B1, does it have a particular neurotransmitter receptor signature? Are there neurotransmitter receptors that are mostly expressed in these regions with take layer 4? And is there something different happening in regions that have a thinner layer 4 or stuff like that? And this is quite a simple question, but oftentimes we encounter quite a couple of methodological issues, because most of the time when we use data from other groups, it's often not the way we want it. Oftentimes we are using a coordinate system that is only for ourselves. So a lot of people work with free surfer, so they have their data in the FS average. Other people use the HCP toolbox, so they have their data in what we call FSLR. So these are different coordinate systems. So when we have, for example, that on the surface mesh that we're using is going to be different. It's going to have a different number of vertices and stuff like that. And our goal is oftentimes to, when we want to compare them, we very much need to be able to merge one coordinate system to the other one quite easily. And sometimes the tools that allow you to do that, because a lot of different tools allow you to do that, are sometimes too complex for a new user that is not very familiar with these complex tools. And so here, what we're presenting today, I'm actually going to skip to the other slide. So the tool I'm presenting today is Neuromaps, which is basically a toolbox that allow you to do exactly what I've just described. So map between different coordinate systems very easily. So it's a Python toolbox, and it's very lightweight. So you don't need a lot of knowledge. You don't need to learn a new environment at all. Well, for those of you who are familiar with Python, obviously, those who are less familiar, you'll first have to learn Python, which is sometimes a daunting task, I must agree. And the second toolbox I'm going to present today is called Abogene, which is also from Ross Marcello. His idea was because he wanted to access the Allen Human Brain Atlas microarray expression data. So I don't know if you are familiar with this data set, but it's basically it's basically the work from the Allen Human Brain Atlas, who gathered 6 donor brains post-mortem and then probed it using microarrays to gather information about the cmRNA expression across the whole brain of these 6 donor brains. And this led them to develop a big data set of more than 20,000 genes across more than 3,000 probes, I think, that give us information about mRNA expression across the whole cortex. But using this tool, using this data set, acquiring this data is sometimes a daunting task because there's so many information to handle. And so what Ross developed is a Python interface that, again, the goal here is always to make it as much easy as possible. And I would say it's quite easy to use Avigene, as I will show you later on. So I'll right away start by talking about a bit more about what I mean by Neuromaps here. And so Neuromaps, how it's been developed is basically as a workflow where at first you start with a brain map of your choice in any type of space you want. So it can be volumetric space, it can be surface space. So that's the one I'm showing on the rightmost left here. And then the idea is that you want to compare this map to a bunch of other brain maps in the literature. And so the first thing you'll have to handle when you use Neuromaps is transform your brain map to whatever brain map, to whatever coordinate system that the people in the literature have used to do their own research. So Neuromaps, one of the first thing it does, it gives you these tools to do these transformations. So for example, from MNI 152, you can transform your brain maps to FS average, FSLR, and CIVET. And then the second thing that you want to do when you have those transformations done, you want to compare your maps to other brain maps from other groups. And this is why we've acquired a bunch of data sets from a bunch of different groups that had made them previously available. And we collected them all and made them into a library that can then be used directly through Neuromaps to do your analyses directly there. And we also incorporate what we call spatial nulls. Well, many people call them that, not just us. And these spatial nulls are super important to do to do comparison between brain maps, because the spatial autocorrelation in brain data is oftentimes creating a lot of problem when we want to compare some things together. So we get inflated p-values, for example, and by using spatial nulls that preserve the spatial autocorrelation of your brain maps, we get p-values that make way more sense to acquire the significance of some relationships. So I'll go over a bit more what I mean by those transformations. So the idea, as I was saying, is that we start with a brain map in MNI 152 space. Let's say, for example, cortical thinning in schizophrenia patients, and then want to compare it to something like cortical thickness, right? T1, T2 ratio, or even like the main axis of variance of gene expression in the brain, or some more exotic map like allometric scaling. This seems like an easy task. Well, I mean, for people like, and conceptually, you might think it's easy, but then when you try to use this data, you realize, well, the cortical thickness and T1, T2 ratio, if you take it from the HEP, is going to be an FSLR surface. PC1 gene expression, it's very likely it's going to be an FS average if you project it to the surface because you've used FreeSurfer, for example, and some other research is done using the CIVET atlas, which is different from the other ones. So now what we want to do is transform these different brain maps to one single coordinate systems. So what we provide is tools to do just that. So depending on whether you want to transform from volume to surface, it's going to be different than if you want to transform from surface to surface. If you want to transform from volume to surface, what you'll use is what we call a registration fusion framework. Whereas for surface to surface, we'll use what we call multimodal surface matching. So, so for registration fusion framework, I'll just explain quickly like how this works. So basically the idea is that we oftentimes like are able to map from, like we've developed the tools to map from a native image from like a single subject to either MNI 152 space or FS average. So these are the template space that are used to just standardize your data across multiple subjects. So the idea here to do a mapping from MNI 152 to FS average is basically to take these two mapping from a single subject, so from native to MNI 152, and then from native to FS average, and concatenate the two to ultimately get a mapping from MNI 152 to FS average. And to like create this mapping, we actually needed to use like a bunch of data. So what we've done is we've relied on data from the HCP 1200 release. And so using this data, we've did exactly this to get a very nice mapping from MNI 152 to FS average. And the code to see like how we've done this mapping is available in a development repository. Oh, and by the way, I just forgot, but I have a GitHub repository for this for this lecture today that you guys can access to have the slides and the tutorial for the second hour. So it's on my GitHub. I can actually show you. So it's right here. So this is on my GitHub account.
I have a small question.
Yes, yes.
You showed the different spaces here, but I wonder what MNI52 version you're referring to. Do you support multiple ones, or is this a specific one?
Yes, so we're supporting two main ones. So we're supporting the 6 linear, the alinear 6, no, the 6 linear asymmetric one, and the 2009 C1. The mapping here was done using the 6 asymmetric one, so the one that is provided from the HTTP. So to keep it consistent with the HTTP tools that we're using to do these different transformations.
And for the pre-server, it's the latest one, I assume, the FS average. I think you also have two versions, but yeah. Okay, cool. Thanks.
When you do the co-registration between different maps, this is totally automatic procedure, or the user can tune the co-registration?
No, so these are automatic procedures for that toolbox. Like
no chance to interact with the system to improve.
No, exactly. Unfortunately, no, not at the moment. But this is good suggestion actually. But yeah, so because they take a while to generate these mappings, because we go through like every single one of the HTTP subjects to make sure that it's like that it looks good.
And do you have a quality control procedure to see how it was?
I'm not sure, because it's Ross that developed that did all of this mapping, so I'm not sure like how he made sure that it looked good. But yeah, I'm not sure about that, but I'm assuming probably. But yeah, sorry.
Are you asking about manual intervention in the case of failures?
Yes.
Okay. So if it dies, do you have any choice to say, no, you went too far here, start there and let it go again?
Yes, because there are some images that are too noisy and the automatic procedure is not so good.
Yeah. But the idea here is that this registration fusion has already happened. Like we have so what we end up with is basically a mapping between the two that we saved and that is available to people, but it's like it's done. It takes a while to do, so it's not what people are gonna do. Like it's already ran. So this is in the code to do that is in the development GitHub repository, but you don't have to interact with this development repository like on the official like Neuromaps repository, like the actual code itself. The mapping is already done, so it's super quick to get from MNI to FS average using this mapping. So yeah, so the idea is exactly so because yeah, so it's not a mapping from native space to MNI or FS average. It's a mapping from MNI 152 to FS average. So we only need one mapping that doesn't change regardless of which patient you're using. So it's basically the idea is that this toolbox has been created so that you kind of compare averaged maps across a bunch of subjects. So it's not for subject specific, really. It's about like if you take all the maps in the literature that have been generated, you want to compare your results to those maps and not to subject specific maps. So that's why the idea was.
So the potential points of failure are for individuals from their native space.
Yes, exactly.
Once they're successfully in that space, then you have these fixed transformations that are similar.
Yes, exactly. Exactly.
So the mapping can mean many things, depending on the number of degrees of freedom that you are allowing, from linear rigid body affine all the way up to very, very high dimensional mapping. And of course, that's problem specific. One has to define what you declare to be real mapping you want to get rid of in the mapping function, real variability you want to get rid of in the mapping, and that which is subject specific, and that's just noise. You can't handle the fact that two brains are not diffeomorphic. So how do you handle this choice of the number of degrees of freedom in the mapping function?
Yes, so I'm not totally sure about that. But I know it's not like I'm not totally sure. So as I was saying, so these mappings were generated by Ross before so it was generated by Russ. I didn't have much control on that side. All I know is that there's no control on the parameters you're using for the mapping. It's done. Like, I mean, it's available, like the information I think is available on the in the paper, or it should be available somewhere. If it's not, please let me know. Like, if you guys like encounter some issues when you read the documentation and you don't find the information, let me know, because it's a great question, but unfortunately I don't know. And it's like, and there's no, like, you cannot change those degree of freedoms. It's one mapping that has been created, as I was saying earlier. But it's a great point that like we should allow some variability like in the mapping. So yeah, so to continue, so if we want to, so this was from MNI, so from volumetric to surface. The other transformations that we can do is basically from surface to surface, which is a different process, where in this case we're using what we call multimodal surface matching. So the idea is that when we have two surface meshes from two different coordinate systems, you want to map them so that some descriptor of brain structure and function is aligned quite accurately between the two maps. So the idea is that if you have two brain maps, two surface mesh, what you have, JRI and SOCI, for example, this is like the basic example, that may not be aligned if you map them together. So the idea here is that to do multi-surface matching, you take some descriptor of brain structure and function from both the source and the target coordinate system. So the main example is so-called F, for example. And the idea is you map both of these surface mesh to their spherical representation. And then what you do is you align the two spherical representation of these surface meshes in the two coordinate systems, such that whatever descriptor you're using, so chi-depth, for example, is aligned on the two maps. And then you use the rotated surface mesh that is in spherical format, and you projected that, and you use that actually to do the transformation. So I'll kind of explain to you with a demo, because like, well, not a demo, but a little figure here so that you guys can understand, because in words it can be sometimes confusing. So yeah, the idea is if example, we want to map from FS average to FSLR, what you'll take basically is some so-called F information from the two maps. So here on the left, we have FS average, on the right, we have SLR. And the idea, we project those maps to their spherical mesh, which are usually the final. So we have this information from the HCP. So from FSLR, we have the spherical mesh that is already available to everyone. And we have the same thing for FS average through resurfer. And then the idea is so if we look at the so-called F information, we actually see that it's not perfectly aligned. So this blue thing here is not aligned with this blue thing here, which like are the same thing. So the idea is that so as I was saying, basically, we want to align these two different things here. And the way we'll do that is we'll rotate this sphere so that now these two parts are aligned together. And then this spherical mesh is then what we'll use to do the transformations from one to the other. So what we'll call it is like the FSLR deformed to FS average spherical mesh. So just as an example, now the two are on the same position in the sphere. So the idea is like if we take some like density map of a GABA receptor, what if we want to convert it to FSLR, what we'll do is project it to its spherical mesh in FS average, then simply project it to this FSLR deformed to FS average spherical mesh, and then we project it to the FSLR mid-thickness mesh, for example. So now we have a mapping from these two that respects the JARA and SOCAR, for example, information of the two maps. So these spherical mesh, so this one particularly already existed. So this deformed to FS average spherical mesh already existed by the people who actually developed this multi-modal surface matching algorithm. But what we added here in our case is we added a spherical mesh deformed to CIVET. So basically, we now allow these types of transformation with CIVET. So this is one of the things we've added, which can be quite useful when you do your analysis. And so this leads us to being able to handle these different coordinate systems. So basically, these are we think it's the most common coordinate systems that people use. So we have FSLR, FS-average, CIVET, and MNI 152. And we have them in different density and resolutions. So we can actually like downsample or upsample our maps if needs be. And what about the big brain? Because like, I mean, this is HighBall, so it's about also big brain data. So it must be known that like multimodal surface matching also exists for the big brain. There's people that worked on that throughout the consortium. So, and there's actually like Casey Pacola, for example, developed this big brain wrap support that allow you to use this so people have created. So here there's this idea that like they've created the same spherical transformed spherical mesh for the big brain that allow you to map, for example, from big brain to free surfer or from the HCP to FSR. And this is very easy to do using big brain wrap. Well, using, so here it's actually a small example. So there's actually a guide, very simple to explain to you how to transform from the big brain to either FSLR or, well, in this case, it's FS average. So I don't know actually if there's, yeah, so this is an example from big brain to FS average, and it's the same idea that I've just explained. So yeah, so the other cool thing about Neuromaps, like aside from this transformation that I've just explained, is the library itself. Because as I was saying, we kind of gathered a bunch of data sets from different groups. Some of them were available, some of which were not, that are like only accessible to Neuromaps. And I think these are perhaps the coolest because they're not as well known as the others. And I'll just go over a couple of them. So like the first one is probably like, you guys are probably familiar with the HCP and some of the microstructural properties that they've shared with the world. So these are group average, T1, T2 ratio, and cortical thickness maps that you can compare your data to. There's also a lot of metabolism maps, so from two different groups. So there's Ted Satterthwaite that shared some of his data, as well as this other group from Marcus Reichel, if I remember correctly, I hope so. And there's also a lot of brain maps related to functions. So for example, the first one, intersubject variability, is about how variable functional connectivity is across subjects depending on brain regions of the brain regions. There's also neurosynth stuff. So for those of you who are not familiar with that, it's basically a bunch of it's a repository of relationship between specific words and activations in brain map in the brain. And so here we have the first component of a big data set that basically consists in a like it's basically the first component of the matrix that consists in hundreds of keywords associated with certain emotions, feeling, tasks, and so on. And each brain region, and what we did is basically computed the main axis of variance across all these different terms across the brain, which gives us an idea of how different brain regions are involved in different tasks across the brain. And it gives us a gradient, which we might call, that is actually very similar to the functional gradient. Well, I mean, this information is quite well known, but like it's actually related to the functional gradient. So we have those two here. We actually have this, basically this functional gradient from Daniel Margulis. We also have a lot of electrophysiology data. So this is again from the HCP MEG dataset. So we have the different powers. We also have this intrinsic timescale map, which is from our group. I mean, I don't have it here, like, but it's from Golia. Well, she used to be from our group. Now she's with Ted Satterthwaite. But it's yeah, so it's from Golia Shafi for those of you who know her. So, and then there's a lot of expansion related maps. So like across evolution, across development, and allometric scaling is like, how does it vary across individual, like the size of the brains, for example. And we also have genomics. So this is using the Allen Human Brain Atlas. We took the main axis of variance across all the genes that they made available to get like an idea of how variable gene expression is across the brain. And so these are all pretty much openly available, unless I'm mistaken. So they're available elsewhere. That's what I mean. But this one is actually available through Neuromaps. So this is work from our group. Well, I mean, it's work from many, many, many groups. But Justine Anson, basically a colleague of ours in Bratislav's lab, gathered, like, just asked a bunch of people if they were willing to share their data. So she just asked them if they wanted to make it publicly available. Most of the time people want to do that, but they just don't know how. So she helped them do that for them. So she gathered all of this data and created this data set of PET receptor maps. So basically a bunch of people had made PET experiments with using tracers to gather information about the density of different receptors across the brain. And so Justine called them and asked them if they were willing to share their data. And what that led to is basically this nice data set of PET tracer density maps across the brain. And so, as I was saying, it's not us who gathered this data. So here I have all the references. I mean, and we have them. I'm going to show you in the tutorial, like, that all this information is available on the toolbox. And like, please, if you use this toolbox and this data set, like, cite the original author, because all Justine did was like ask them to share the data. She's not the one who actually used this data. So that's pretty much what the library is all about. Aside from that, we have this third thing, which is quite important, I would say, is this idea of spatial nodes. So as I was talking earlier, so the idea of the whole neuromap thing is to be able to contextualize, or is also compare our brain maps to the brain maps of the literature. And the way we do that, once the brain maps are transformed and we are able to actually do a good comparison, is basically it's an example, of course, we could just do a correlation. Then we get we have these bunch of brain maps here, and then we correlated them with our source map, and then we get a bunch of correlations. But like, the question is, how do we evaluate the significance? Because as I was saying earlier, like, there's this thing that is called spatial decorrelation. So there's, like, brain maps are smooth, basically. And as a result, there's a lot of patterns in the brain where, like, many brain regions have a similar value, whatever it is. So like T1, T2 ratio is going to be similar for brain regions that are close to each other. Thinning is going to be similar for brain regions that are closer together, and so on. And that leads to a lot of problem when we want to do comparison between brain maps. And this problem is basically that the p-values we obtain when we compute correlation, for example, are going to be inflated. So this is just an example. So this is just using random Gaussian fields. We generated here, for example, 2 brain maps that are completely uncorrelated. But as we increase the smoothness of this map, what we see is actually the correlation gets pretty high quite quickly. It gets from 0.00 to 0.9, which is, and even though, like, these brain maps, like the actual, like the original brain maps, were not related at all, there was a correlation of 0.00. So this is just an example of the big issue that happens when you want to correlate brain maps together. And actually, like, I'm actually like showing this again in different forms. So here we can actually see that what's happening is that when we look at the correlation between pairs of random brain maps, as we look at the density of correlations we get for a random sample, what we have is that the distribution gets flattened a lot, and then we have a lot of extreme cases where we have very large correlation, even though it's a completely random pair of brain maps. And the big problem with that is that we end up with a very large false positive rate, where if we have a very highly spatially autocorrelated brain maps, you might have like a false positive rate of almost 1, and that any type of correlation you get are very likely to be significant, which is not what you want when you do these types of analysis, of course. So basically what we need to do is like develop new statistical tests that allow us to control for spatial decorrelation. And the idea is to use permutation testing, a very simple process where basically we take the 2 brain maps we want to correlate, we get a correlation. So for example, minus 0.65. And the idea is we want to generate null maps from the dependent brain map that replicates some properties that we want to control for. In this case, it's the spatial intercorrelation of our brain map. And so the idea is that once we've created those surrogate brain maps, we'll again correlate our original result and get correlation that would be set as our null distribution. And then what we want to do is compare our empirical result to this null distribution to get a more accurate p-value. And then we can simply compute the p-value. So here in this case, we can get something like 0.06. And so this is all good, but how would we generate those surrogate brain maps? There's a bunch of different ways to do it. I would say that, I mean, of course, there's the naive models that don't preserve special correlation. Like if you could simply use a student distribution, for example, or use a permutation test, but without any type of control for spatial correlation. In that case, it's not, I mean, I wouldn't suggest it for the reason I've explained. You would get inflated p-value. The thing you could do, however, is generate them using what we call spin test. I mean, we call them here spatial permutation models, but some people have called them spin tests in the past. You might have heard of it before, but the idea is that you take your brain map. So for example, we can see like the little idea here. You take a brain map, for example, in FS average, for example, and the idea is you project it to its spherical surface mesh. And then the simple idea is you rotate it and then project it back to the original like mid thickness, for example, surface mesh. And so what you end up with is basically a brain map that preserves all of the spatial correlation in the original map, but that disrupted all of the relationship between specific regions of the brain. So that leads you to, if you want to compare it to another brain map, you would likely get much smaller correlation in the case if the pairing between the two is completely random. So this is one way of generating surrogate brain maps. The other way is using parameterized data models. So these are basically, the idea with these models is you basically develop a measure of the spatial, you develop a technique to compute the spatial correlation in your original brain map. And then you, using whatever algorithm you want, well, I mean, there's specific ways you do them, but using an algorithm, you then regenerate null models that preserve the spatial correlation score that you've computed. So Joshua Burke, for example, created 2 different versions of this parameterized data models, and there's also the model that relies on the Moranzai measure, which has been developed by Reinder Vazdeville from Boris's Bernard group. So like then the question is like, which model do we choose? Because I mean, I've just talked about how there's different models available. Well, the answer is it depends what you want to do, because like every single one of these models have their own caveats and their own particular things that allow you to either like them or not. Of course, you shouldn't use naive models. That's the main takeaway, I guess, you could have from this presentation, because they give you inflated p-values. The thing about spatial permutation models is that, of course, they require surface-based data because you have to project your surface mesh to a sphere. I mean, you can transform your MNI, your volumetric data to surface using Neuromaps, but sometimes you want to keep like the original representation. Yes.
Sorry for interrupting because I need to leave, but I want to ask you a question. Yeah. Do you know the SERFstat by Worsley, the SERFstat model software?
Yes, yes.
How does this software compare to SERFstat? So do you already incorporated random field theory, all the linear mixed models, all the things that are incorporated in SERFstat?
Yeah, we do incorporate. So I'm not too familiar with everything that's available in SERFstat, but like they do have null models that are very comparable to them. Like they do have spin tests, they do have Moranzai at least. They have statistical tests that are very similar to what we have here.
This has more or less same functionalities as SERFstat, but more modern statistics.
And there's more null models here. They don't have all, like they only have one variation of the spin test. For example, here we have 5, and don't think they have all of the different. I don't think I'm not sure if they have all the BERT 2018 or BERT 2020. I know they probably have the Moran because they've developed it. Yeah, but they do have more statistical analysis tools. So here we're just like the main idea, like we're not offering a lot of tools to do the statistical analysis. That's more what they do. Here we're mostly focused on giving people access to those null models. So as we say, we have more than them. and they have, but we don't have the statistical analysis that they have. So you might want to use both. Their ones to use the statistical analysis, and ours if you want to focus on specific models.
Thank you very much.
Okay. Yeah, so parameterized data models, by the way they're defined, we can actually use them both for surface and volumetric data. So this is quite useful in case you need to use volumetric data. And yeah, so like the question is, okay, so all these different spatial permutation models, what are the differences? Well, some of them do what we call imperfect permutations, in the sense that we want sometimes to generate. So the idea is basically to preserve the actual like full permutation. You sometimes have to do a lot of work, because you want to, when you rotate your sphere, basically. Oh, I should have said something beforehand though, but I'll skip to this here before. So the idea here is that one of the things that differs between all these different models is that some of them work at the vertex level. So, but most of them actually work only for parcellated data. And so basically the only one that works for vertex level data is the Vasquez-Rodriguez, which is also the original one from Alexander Aaron Bloch, who developed this idea of spin test. And this one works. It's basically the one I've talked to you about, like you take the preserve for vertex map you have, you project it, you rotate it, you projected that. This is the original one. And this other one are alternative models that only work for parcellated data. And the idea is that there's some issues with some of them that, I mean, are not issues in the sense that like if it might be something you want, but like in this case, in the Baum and Kornblad version, they when you spin it, which is actually something that actually happens also for the previous one, is that you might end up with your medial wall inside of your brain map. Sometimes when you compute correlation and you have a null distribution, it's not a big deal because you just like ignore those values when you compute the correlation, but sometimes it's a big deal. So depending, you might want to not choose these models and use ones that give you complete permutations. And there's also ones that give you perfect permutations. So these three models, when you permute your brain maps, you're gonna assign, yeah, so it's not, so here, imperfect permutation for the Vasquez-Rodriguez model in case you're using a parcellation. If you use vertex level data, it's gonna be perfect, but if it's parcellated data, yeah, I don't know what I just said, but if you use parcellated data, it won't be perfect. That's what it's saying here, basically. And if you want to have perfect permutations between, like from your original brain map, you want to use those other two. But the issue is that these take way longer to do. That's actually why they're not available at the vertex level, because you have to kind of use algorithms that match each parcel to their closest representation in the original brain map. So this is troublesome to do, so it's longer to do. So that's why it's only available at the parcel level, so when you need a parcellation. So yeah, sorry about this confusing moment, but all the information is actually also in the documentation. And one last thing I want to say is that for the parameterized data models, in theory, they both work for parcellated or voxel or vertex level data. But you must be aware that if you do these models using voxel or vertex level data, it's very complicated in the sense that it's very much time consuming. It takes about like an hour, a day or two to run, depending on the size of of the density of your map, and it also takes a lot of storage to store those maps, obviously. So like it can be hundreds of gigabytes of temporary storage. So this is something we're working on to make them more usable, but so right now, like it's not super easy to use these parameterized data models at the voxel vertex level. So we usually recommend people using a parcellation to do that. So the question is it worth it, right? Because I just want to show here basically that if we look at, if we looked at the false positive rate that we obtain with these null models, I had shown earlier during this presentation, like the spatially naive line here that basically shows that if you have spatially intercorrelated brain maps, you almost have a false positive rate of 1, in the sense that all of your correlation are going to be deemed significant, even though they're not. But if we use those null models, we can actually see that it's not as bad as it used to be. And by that, I mean that there's still some p-values are still going to be slightly inflated. We don't get like a false positive rate of 0.05 as we want, especially for highly spatially autocorrelated brain maps. If we have like slightly less spatial autocorrelation, it won't be as bad. And it also works when you use parcellated data. So this is when we use the Cameron atlas and the Schaefer atlas. So yeah, so like the main takeaway is basically, yes, they do, these spatial nulls do work. The false positive rate drastically increased. And there's also a little thing that we might want to show here is that the parameterized null tend to perform not as good as the spatial permutations. So I don't like, so this is something we've observed. So this is like not, so this is actually coming from a paper from Ross that actually investigated every single one of these different techniques to generate a spatial nulls and evaluated how they are accurate or not. And so it's a very thorough paper. So if you're wondering like which type of spatial model to use, you might want to refer to this paper where they describe every single one of them. And yes, and so the final part of Neuromaps is basically to just do the enrichment analysis, right? So like we have these correlations, and then we get p-values using these spatial null models. And what we see is basically like you have a bunch of correlation at the beginning that some of them would have been significant, but what we end up with is only 2 significant relationships between cortical thinning in schizophrenia and other brain maps that exist in the literature. And these are related to allometric scaling. So like how variable the scale of the brain is across brain regions. So yeah, so this is an example of analysis you could do, for example, with Neuromaps quite easily. And yeah, so for Neuromaps, that's it. So it's actually available as a Docker container, so people sometimes like that. And you can also install it directly if you want using pip or using directly like from our GitHub. And there's a GitHub repository for that. I'll show you on the second hour. So feel free to let us know if you have questions or issues directly on GitHub. You can also email any of us who would be very happy to answer your questions. And there's also a very thorough documentation explaining every single function that exists in this toolbox as well. And Justine, I have to say that like Justine is one of the main person that worked on this toolbox alongside Ross. She's the one who wrote most of the documentation. She gathered, as I was saying earlier, like most of the data that is available in there, as well as Ross, of course, who actually built the toolbox. Me personally, I'm the maintainer, so I'm going to be the one that's going to answer your questions. I'm going to be the one who will fix whatever bugs you'll find, because you'll find bugs. We do find bugs, but we fix them quite rapidly. At least we try. And so, yeah, so I'll be more than happy to talk to you guys after if you have any more question about Neuromaps. And then I'll just quickly, I don't know when I started actually, but just quickly, I'll talk about Avegin. So as I was saying, Avegin's goal is to allow you to fetch and work with the Allen Human Brain Atlas data more easily. And so for those of you who are less familiar with this data set, is basically a data set of microarray expression data that has been gathered, as I was saying earlier, from 6 post-mortem brains. And what they did is they ran a microarray analysis and obtained cRNA probe that they used then to get RNA expression from more than 20,000 genes across like more than, so basically across more than 3,000 sampled tissue pieces from these post-mortem brains. And so what that leaves us with is basically a repository, a data set of more than 20,000 genes where we have expression of them across the brain. Well, not full expression across the brain, that's what I'll get to later, but some expression for some regions of the brain. When we use this data set, actually, one of the things that like might be troublesome when we're not familiar with it is that we have to pre-process it. Because as I was saying, not all the genes cover the whole cortex. So when we want data, one of the main tasks when we use this Allen Human Brain Atlas data set is we have to pre-process it, because like not all gene expression is available throughout the cortex. This is just an example of the things that might be problematic for you when you want to use it. So there's So there's Arina Avnatkevich-Ute. She actually like developed this whole framework of different things you have to do to use this data set in a correct way, we would say, because there's a lot of caveats that can happen if you don't. And these processing steps involve many things. So for example, there's the simple idea that like the matching between mRNA data to actual genes is sometimes not up to date using the Allen Human Brain Atlas. So you might want to use more up-to-date information to match your mRNA things to your genes. There's also this step of like doing actual data filtering. So sometimes like the mRNA data you have is quite noisy, so sometimes it's not usable. So you might sometimes want to exclude some probes that are not exceeding background, for example. And when you have multiple probes for one single gene, you might want to only pick one that is the most accurate in some way. You also want to decide like the tissue samples that you've used to get this information. You want to try to figure out where it's actually in the brain. So there's actually a question there of what you want to do. You also want to normalize your data across subjects, because if you look at the PC1 here, the idea, the PC1 and PC2 of like expression data, you actually see that we can actually take out like each subjects, each one of the 6 brains. So what you want to do is normalize across subjects to get like information that is reliable across subjects. And you might also want sometimes to filter out some genes that are not consistent across the individuals. So if you're not consistently expressed across different individuals, you might think that this gene is not accurately like captured, like the probe, there might be noise in it and it might not be super useful to you. So you might want to filter out those genes. And ultimately, what you want to end up with is a brain region by genes matrix that tells you the expression level of each gene in each brain region. And this is quite a daunting task to do all of this processing. And actually, that's where Abogen came in, because we've actually added all of these processing steps inside of Abogen that allow you to just decide. And in this case, you can actually decide how you do the pre-processing as opposed to Neuromaps. You'll get to decide every single one, or every single one of those steps, what you want to do. And this is super useful if you want to gather this data and don't have to handle a pre-processing yourself. And yeah, so I must also say that like if you want to access this data like just by itself, it's actually available. Like, you know, you can download it from the website, or you can use their API, the API that also works. And that's actually what we're using to fetch this information and everything. But really, I would say that the most useful things about Neuromaps is all of the pre-processing that is done that allow you to just end up with a gene expression by brain region map that tells you the information you actually want and need. And yeah, so what I mean is as simple as that, like with one simple function call, we get this brain map, we get this matrix, sorry. Like all we need is a parcellation that tells the function which are the brain regions we want. And we can also use a brain mask that actually returns all the tissue samples, like that returns expression for all the tissue samples that are within the brain mask that you want. And it can do more funky things. So it can actually like generate a densely interpolated expression map. So like, because I kind of talked about it earlier, but like you have tissue samples of the brain. So that means that like you don't have a full brain map of expression across the whole brain at the vertex level. But what we've added to Abogen is tools that allow you to interpolate your data so that you can obtain a densely interpolated brain map. So that you can use it for like your full brain analyses in case you want. And we also have multiple methods for missing data. If you have parcel data, you'll either like can do like centroid matching or interpolation again to add information to the parcels where you actually don't have gene expression information, but so that you can at least use these full brain maps to do your analysis. And yeah, as I was saying, Abogen handles the various processing steps for you. And what's cool about it is that it might get confusing because there's a lot of these processing steps, but what it returns you, if you ask it, is going to be a full-blown like text that actually describe all the methods, which you can just simply take and copy and paste into your report so that the other people actually know what you've done in detail, and so that your experiments are actually replicable. So that's actually, I think, is pretty cool. And yeah, so Ross actually has a paper about his toolbox where we actually also evaluated the usefulness of each one of these processing steps that I've described. So you get to see like which one are most important, which one affect the most your data, which can be super useful in case you want to deal with this data set. Yeah, so that's pretty much it for the first part. So I've kind of explained what these toolboxes are about. And then I don't know if I have like a second part of how this works or.
Yes, so we can have a few questions now and then we can take a short break.
Yeah.
For you to fill up your water bottle and then we can continue with the hands-on part. So any questions for Vincent? I see that you have a very particular case of using multi-resolution data in this project.
Yeah.
From very, very sparse allen brain samples to the, coarse resolution PETs to then the MRI and fMRI data that are also involved in there. How do you deal with this large differences in resolution when you look at maps?
Yeah, it's a great question. Well, the idea when we do these analyses is usually, and we've actually like implemented it somewhere in the toolbox, you'll see, but our default is always to downsample as much as possible, obviously. So in the case, so that's why we actually encourage people to use sparse relations as much as possible, because that's when like the issue of like multi-resolution data is going to be the least problematic. So we usually like encourage people to downsample the data as much as possible, so that especially if they want to use things like averaging, which is perhaps the coarser resolution you have. So yeah, that's how we handle it. Like in most transformations, if you don't ask for it, they're going to downsample it to the lowest resolution that you have. So for example, if you have a map that is in FS average 10K vertices, and you have like a very high resolution of like 164K vertices, it's going to downsample it to 10K. Yeah, that's.
So let's take a short break.
All right, so welcome back, everyone. So in this second part, I'll just so in the first part, I was kind of like doing a slide presentation and just going over everything, but now I'll just show you the actual tools themselves. I'll show you the documentation, the GitHub repository, and a bunch of different things so that you can get familiar with all of the stuff I showed. So we can start actually by looking at the documentation for Neuromap, I would say, because that's where most of the information you'll need to use it is going to be. So basically, the first thing you'll have here is the installation requirements. So if any of you want to follow along, I encourage you guys to do it, and you can actually just download it. The best way to do it is through GitHub. So, through Git, using our GitHub address here, which is actually this repository right here. That stores all of our code, and that you guys can download. And then you can install it using pip. This will allow you to follow along if you want later on. And so, yeah, so this documentation actually has information about installation and setup. We also have a couple of user guides. So like if you want to know more about like this coordinate systems, the different ones we support and what they are actually. So here, like, as I was saying, so we're using the nonlinear sixth symmetric template. Yeah, exactly. But we also have the MNI 2009C, but it's not written there. Then all the different coordinate systems, and like these are examples that I'm going to actually show later on. Then there's a bunch of different other user guides. There's a couple of examples. And there's a reference API as well, which I'm going to use for the presentation today. So I'll go over some code and I'll refer back to this API where you can actually have some information about what is actually going on and what like all the different functions that I'm going to use are. So yeah, so I'll start. So I'm going to use Spider. So it's so I'm not going to use Jupyter as most people when they do tutorial do. I'm gonna simply use like Spider and like IPython to run stuff in front of you guys, and the data is gonna appear like on the top right as in MATLAB, for you those of you know MATLAB. So I'll be able to dive into the data itself and show you guys what is happening. So the first thing I'll do is actually like just import stuff. This actually I have to change that. So I've set it so that it's in the current, so that the repository is in my, oh, no, actually, yeah, okay for me. But you guys, if you want to follow along, you want to change this current working directory to wherever you downloaded the tutorials. But so what I'll do, I'll first start by doing this. So I'll import everything I need. And by the things I need, I mean very simple stuff. So like Nibabble. So this is the main toolbox that is in Python to use if you want to open or close neuroimaging data. It's super easy to use. I'm going to use it a couple of times in this tutorial. And like aside from that, it's simply like NumPy just to play around with the data itself, and like matplotlib because I'm going to show you a couple of results. And aside from that, I'm importing Neuromaps, sorry, of course. And in this case here, I'm importing a couple of functions I'm going to use later that allow you to handle images quite easily. And these are like super useful functions that like are usable even for you, like in your other projects. You don't have to use Neuromaps by itself, do all the things I've showed. You can simply use those functions that allow you to do cool stuff. Like for example, transforming annotation, annote files, so from FreeSurfer to GIF images, for example, or relabel GIF. Well, this is going to be more complicated to explain. I'll show you later what it's used for, but you can also convert delabel. So basically SIFT images to GIF images using these tools from Neuromaps. So now that it's imported, kind of show you what we can do with it. So we can access first the data sets. So I'm importing the modules related to that, and the documentation, of course, is on the GitHub. As I was saying, the API is right here. So what we can do basically, so the way like Neuromaps work is that we can use this function in this module to fetch all of the data sets that we have available to you guys. So one of the, there's two different types of things we can use. We can use atlases and we can use annotations. So for atlases, we can simply do like data sets that fetch annotation, and then we'll get all the atlases we have. For example, in this case, I've set it so that I want MNI 152 atlases in 1mm resolution. So now you have access to all of the atlases that I have in MNI 152. So we have the 6asym and the 2009C asymmetric versions of MNI 152, and we have like T1, T2, T1 weighted, T2 weighted, white matter, gray matter, CSF, and so on. So these, you guys can just use them for whatever reason you want afterwards, but we use those atlases as well for doing a bunch of different things that you won't be aware of. And there's also, we can actually do the same thing for example, FS average atlases. Again, we have all the different surface mesh. Among them is the sphere, which is important for like generating null brain mass, as I was saying earlier. And so, yeah, so this is to fetch atlases, quite simple, but perhaps what's the most interesting is the annotations. So I'll just go to this web page here where we actually can see what all the annotations are. So it's right here. So we can access this through the GitHub page. So it's in the Neuromaps. You go here on the wiki section, and then you click here to get all the information. Then here again to get this Excel spreadsheet where we have all the information about everything that's available to you to use as annotations. So the way the name of the annotation is designed is basically we have first the source of the annotation. So it's going to be who acquired the data first. So for example, for this particular map, we'll have Beliveau 2017. So he's the person, Vincent Beliveau is the group that acquired the data. And so the first thing here is the source. So it's his name, then it's SIMB36, which describes a bit what the data is. So this is a PET tracer for a serotonin receptor, so 5-HT2A. And then it tells you in which space it is and the resolution. And so this is how every single brain maps that we have available are described. And then here we have in this Excel sheet information about what it actually is, so that you can actually know that SIMB36 is a PET tracer for those of you who don't. There's actually information about the size of the data set, because these are average brain maps, so we don't have, for consent reasons, we don't have access to the individual maps from every single subject. So these are group averaged, already projected to a particular atlas. So these are in MNI 152 space, but we have information about how many people were used to generate those group average maps. And then, very importantly, we have the references. If you want to use those maps, please cite those references because that's where they're coming from, not from Neuromaps. So yeah, so this is information about the maps and how do we fetch them. I have to say that we can actually, oh, my thing here is actually hiding me the slides. Okay, so here, so if you go on the OSF repository, that's where all of our data is actually. So we have annotations here. We have all of the different sources, and we can actually I'm pretty sure we can download them directly through there. Like let's say click here, download a zip. So yeah, we can download all the annotation like that. And so that's one way of acquiring them. If you don't want to use Neuromaps, if you're not familiar with Python, it's super easy. Well, I mean, now it's waiting. I don't know what's happening, but like it shouldn't be too much of a problem. But if you want to use Python to do all the different things that Neuromaps allow you, you can use different functions. So here it lists all available annotations, lists all of the annotations that are available to you. Again, it's the name that I've described earlier. You can actually fetch them once you know which one of them are available. Then you have all the annotations here just for you, and they are basically file paths that are basically pointing to where the data has been downloaded for you. And I mean, it's worth mentioning that this data basically is downloaded in a repository that is going to be in your home directory. So this is my home directory user Vince. It's going to be in a repository called Neuromaps data. And then that's where all of your data is going to be downloaded once you use the fetch functionalities of Neuromaps. So now we have like NeuroSync, PC1, MNI 152, and then the actual nifty image for the MNI 152 image of this particular annotation. We actually have tags as well. So they're available to you so that you can if you want, you can fetch one specific type of brain annotations for your analysis. So for example, receptors is a tag, so you can just do receptors. It's called to data sets that fetch annotation, tags, receptor. Then you get just the receptor annotations, basically. We have 42 brain maps of receptors. And then again, these are file paths. So yeah, so that's basically how you access all the data sets that is available to you through Neuromaps. Then the thing you might want to do is do the transform that I've described, right? So I'll give an example using the NeuroSynth PC1 map. So one thing we can do is fetch the annotation using data sets that fetch annotation source is equal to NeuroSynth. We have this string here that is basically the path to the MNI image. Can actually show you that it's actually existing. If we go Vince, then Neuromaps data, annotation, then it's NeuroSynth, and we can see that I downloaded every single one of them, called PC1 MNI, and here's the image, and then we can look at it. It's gonna look super ugly because this thing is not super great, but if we change this, we're going to see something. We have a brain map. So this is like, when we're talking about resolution, like this is an example of a brain map that is not super clear, like what's happening. We have data everywhere because this is like just gathering a bunch of data from a bunch of studies. Like if you guys are familiar with NeuroSynth, you wouldn't be too surprised that it looks like this. But this is an example of brain map we have. So like, then if we want to transform it to use it for different things, we can simply use a function called, so this is in the transform module. I'm actually going to show you guys what functions are available to you. If you go, so here is the reference API, this is like data sets, all the functions that are available to you to fetch data. And then let's go to the transforms module, where we have in here like all the different transformation you might want to do. So you have MNI to a bunch of different space, FSLR to stuff, Civit to other coordinate systems, and FS average to other coordinate systems. And I mean, you have every single pairing available. You don't have, of course, surface to volumetric, because when you have surface data, you don't have everything that's not on the surface, obviously. So like mapping from surface to volumetric is not something we recommend necessarily. So we don't have them for you guys, unfortunately, these mappings. So yeah, so now I'm gonna use one of these functions as an example. It's quite simple to use, basically. You just like the GIFT image that I'll get as a result. I just get them by saying transform that MNI to FS average. I give the MNI image and I say which density for the FS average map I want. Then I just run this function. There you go, I had it. So GIFT images, I have my 2 GIFT images for each hemisphere. And these have been loaded through NiBabel. So like we're using a lot of functionalities from NiBabel, as I was saying earlier. So this is just an example. So this is all like the GIFT image object here is defined through NiBabel. And then what we'll do here is just a quick script here I'm going to run just to set the values in the medial wall to none. Because as you might be not surprised about is that when you do this transformation in this specific case, the medial wall is going to be filled with values. And for the subsequent analysis I'm going to run, I won't want that. It's going to be problematic. So I'll set everything to none. And then we can see what it looks like by saving the GIFT image. And I'm going to just show you that transformation actually happens. I mean, it's this one here. I was prepared. So it's actually what it looks like in FS average. So we got like this MNI image, and now it's an FS average. And yeah, and it looks a lot like the functional connectivity gradient from Margulis, as I was saying earlier. Yeah, I mean, there's nothing much else to add to that. So now the next step we have is, okay, we want to compare it to all the different maps we have in our data sets, for example. So actually, I'm going to first generate null brain maps from the NeuroSynth map. So what I'll do is I'll use the Alexander block null model. So again, here, this is in a different module. It's called the Neuromaps.nulls module. I'm going to show you guys all the different null models available. So these are the ones I described earlier during my presentation, but you have like Alexander block, Vasquez Rodriguez, and so on. So these ones are all for spinning the data. So this is for surface data. And these 3 are the parametric nulls that can be used for volumetric data as well. So now I'm going to use the Alexander block 1 for this presentation as an example. Actually, I'm not going to do it because it takes about 20-30 minutes to generate 1,000 models, but I was prepared. So I've actually ran it already. And what you end up with basically when you run this function, which basically takes as input your FS average maps, so 2 GIFT images. You say like in which atlas this image is in, and the density and the number of permutation you want. And then and then the nulls, what it looks like ultimately is an array of float where you have every single vertices in your brain map, and you have 1,000 versions of that null brain map that you'll use to do your analysis later. And I'm actually going to show you an example of what it looks like. Did I load it? I think I wasn't as prepared as I thought. No, I forgot to load it. So I'll do it right now. What like these brain maps look like. So I've saved them as GIFT images right here. So this is for left hemisphere. So it takes a while to load because I have 1,000 of these brain images. There you go. And then we'll see what it looks like in a couple of seconds. There you go. So I'll just show the left hemisphere so that we don't have to load both sides. There you go. Okay. So that's what it looks like. The color map changed, obviously. But you kind of see, I'm actually going to keep the same color map as the empirical one so that we see the difference. But we get something like that. So the spatial intercorrelation is preserved. So the blues are like all packed together and the red areas are all together, which wouldn't be the case if you use a complete random permutation. And what's worth noting is that, as I was mentioning, when you do those rotations, sometimes what's going to happen is you'll have your medial wall somewhere random on your brain. So the idea is that when you'll do your correlation, you'll be able, using Neuromaps, to ignore these regions to get a correlation just between the brain regions that actually have data on them. And this is not problematic because you have it's a null distribution, so the correlation are still going to be pretty accurate even though you do that. So yeah, so these are the nulls and what they look like. So yeah, so this was the code to just convert the null data that was simply like a NumPy array to actual Gifty images. So I used a construct shape Gifty function from Neuromaps, and then I saved them using Nibabel. So yeah, so now I've generated my nulls. So what I can do then is compare. For example, what I'm going to do today is it's not all written. So I'm going to just compare my neurosynth FS average image to images that were originally in FS average, like just because for the sake of simplicity, that were originally defined in FS average in our repository. So there's these ones. So I'm going to use GenePC1 and a bunch of density maps, as I was saying earlier, that we have that are pretty cool for specific receptors, serotonin receptors mostly, and GABA receptors. So we can fetch these annotations simply as we were doing earlier. And then we can actually, what we need to do this analysis is resample, because some of them, I mean, most of them actually are going to be in FS average 164K. So I'm going to resample them to 41K so that they match the transformation I had done before, which was MNI 152 to FS average 40K. And one thing to note is that we're using the convention, like a convention that I think we've created because nobody uses that much, but we're using the number of vertices to specify which version of free surfer of FS average images we're using. But like these are basically what we call like FS average stop, FS average 5, FS average 6. So these are the different resolution I'm talking about when I say 41K and so on. So okay, so it's done. So now we have all our FS average annotations in 41K. And then all we need to do, actually, I'm going to show you an example of what it looks like first if we don't use null model, if we don't use models that preserve the spatial autocorrelation. So what I'm going to do is, I mean, it's already ran for me, so because it's going to take about 2 minutes, which to me, it's a bit too long. Probably this next session is a bit too long to do like in front of you guys. So I've saved the results, but you basically can use stats.compare image. You take your neurosynth FS average image, you take your annotation, and then you set the nulls as being the permutations that you're using. So here's random permutations that don't preserve anything. Then you have to specify that you want to remove the NaN values, as I was saying, like you have in free surfer, not as I was saying that you have like the medial wall that have been moved. So you want to omit those NaN values. And now I'm going to use the Pearson correlation, and then you correlate them all for each one. And actually, I'm actually doing it in a different way here. I'm going to do it manually so that we can actually see all the correlation and the null distribution, what it looks like. Because in this case, what it would return you with is a correlation and a p-value. You wouldn't get like the null distribution of correlation score. So I did it manually just to get these results, which I'm going to load right here. And I'm going to show you like what it looks like if you don't use null, basically. Look, if you don't use null, what it looks like is this. Right, you have null distribution centered around 0. You're like, okay, it's pretty good, cool null distribution. But then when you look at your correlation, you get this, right? The null distribution is very much not far away from 0, and it doesn't go far from there. And most of your correlation you're going to get are not close to 0 at all. So like all of these correlation would be significant. That's not what you want usually. You want like meaningful correlations, meaningful relationships. So now I'll do the same thing with spatial autocorrelations. So as you remember, I've generated the nulls earlier. So now in this case, I'm doing the same things. That's the compare image, but I'm using the nulls, which I had just to not confuse anyone, called them new nulls, which don't mean much. But I should have said like Alexander Bloch nulls, because these are the specific spatial permutation models I've used to generate them. And again, I'll do it manually. I've done it manually so that we can actually see the null distribution now. And now the results look like this. So they're still centered around 0, which is what we want because these are random maps. But now we see that the range that they go towards on both sides is much larger. So that makes the p-values will get much more, it's going to be harder for them to be significant. And indeed, if we look at our results now, we see that like they fit inside of the null distribution way more. And then what we can actually see if we look at the actual p-values, which are in PL, is that we only have one p-value that is significant, and it's the fourth column. So it's this one. So the only significant correlation we find between neurosynth, the map that I used, and our annotations is with the receptor, the density of serotonin 1A receptors. So there you go. So that's basically what Neuromaps is about. I actually have another version of another example here where I'm parcellating the data first. The reason why I'm showing this other example is just to show that like it's much faster to do. I'm actually going to do it quite quickly. So like this is from the parcellate that is from the parcellate module from Neuromap, which has this parcellator class that allows you to parcellate your data however you want. For this section, I'm going to use like NetNeuroTool, which is a different Python module, which is basically tools that we're using in our lab, in the NetNeuro lab, to do a bunch of different things. So now I'm going to use just to fetch the Schaefer atlas and to plot images that I'm going to show later on. And then I'm just have this helper function just to plot images later on. So what I'm going to do is just compare the Margulis gradients to all the receptor maps that we have. It's going to be quite quick. So the first thing we need is the parcellation map. So here I'm importing all of them. So NFS average 41K and FSLR 32K and MN152. These are all available, like these different ones. I'm not doing anything. I'm downloading directly from the Yale related GitHub. So the Schaefer atlas is from SEO groups, and they're available on their GitHub. So I've just fetched them here. And what's worth noting is that, as I was saying earlier, well, I wasn't saying when I mentioned it, but like these FS average parcellation maps are going to be as .anode files because they were generated through FreeSurfer. So you'll have to call anode to Gifty to convert the anode file to Gifty images, which is what we need in Neuromaps. And we also want to relabel the Gifty images because our different functionalities that rely on parcellations assumes that your parcellation are continuous across hemispheres. So like if you have 1000 node parcellation, 500 on the left, 500 on the left, you want your parcellation to be defined as 1 to 500 on the left and 501 to 1000 on the right. Sometimes your parcellation files are going to be 1 to 500 in both. You don't want that when you use Neuromaps. So you have this function called relabel Gifty. And so I'm gonna do this for FS average 41K and FSR 32K, and for MNI 152 it's much easier because I don't have to relabel anything because it's just one image. So I'm importing them all, and then the way I'm going to parcellate is I'm going to create those parcellator objects where I specify which parcellation I'm using in the space in which it's defined. And here there's this actual thing that is called resampling target. It's asking you whether, in case because this parcellation process can be done even if you have maps from different space. So I could use like parcel MNI 152 to parcellate that FS average map. And in that case, the resampling process from SO to get from FS average to MNI would happen in the background. So then they're asking like, which map do you want to resample to which? So here it's not going to be used because we're using MNI maps to parcellate MNI maps. Yeah, it's worth noting. So to do this experiment that I want, what I'll do is I'll fetch the functional connectivity gradient from our dataset module. Then I'll call park FSLR. So this dataset, this annotation is defined in FSLR. Should be fine. I'm almost done. So I'm going to do park FSLR, which is the parcellator object I created earlier, and do fit transform. And then I'm going to ignore the background data. So what that means is I'm going to ignore the NaN values that might be in our annotation because we don't want to use them to parcellate our data. So there it is, and then we can actually look at the parcellated data if we want. Oh, there it is. So right, our brain map is now parcellated into. Oh, God, it closed so fast. So yeah, I'll skip over this example because it was pretty much the same thing, and also because it kept everything. That's great. Okay, so I'll skip over that. Actually, I can do it. So basically, I'm going to parcellate all the data for the receptors. Oh, I forgot to fetch the annotations. There you go. Oh, actually, there's this important thing here that is mentioned that is worth noting, is that when you use receptor maps, transforming from PET images to surface images is problematic. It's much more difficult than if you use like other type of data. So Vincent Bévot and this Norgard, these two like groups actually shared with us their FS average transformations as well, because they've done a lot of work in trying to make accurate mapping from PET images in MNI space, well, in volumetric data to a surface level. So it's worth noting that if you use receptor maps, you have to be careful when you transform them to FS average, and you want to use, in specific cases like these ones, you actually want to use the FS average map directly if you want to use them on the surface, because they've done a very good job at creating nice representation of what they look like actually on the surface. So that's what this warning is about, and it's important to consider that. And so that's actually why I'm going to skip over these Bévot and Norgard maps defined in MNI 152. But then I'm going to parcellate all the other ones. And then the next step is going to be to generate the surrogate maps, again, using the Alexander block model, but it's going to be way faster than 30 minutes because we're using a parcellation. And then I'm going to show you guys the results. So is it done? Yeah, I think it takes longer than I thought. Shouldn't take too long. But actually, I can skip over that and actually give you the results. So what we find at the end is that the significance results are going to be that you have a correlation between the Margulies functional gradient and two receptor maps of norepinephrine transporter. Actually, I have no idea. I just did that yesterday. I don't know what the meaning of this correlation is that is significant, but these are the only two significant correlations we find. And actually, we could argue, it could be that it's not significant either, because like I haven't controlled for multiple comparison, which is worth emphasizing that you have to do that, because I just did like 40 correlations straight. So like, makes sense that you'll have like inflated p-values as well. So oh, the import failed at some point. Anyway, oh yeah, because I didn't run it earlier. I guess I can do it just for the sake of finishing, because I'm done after that. So I have imported it. Where was I? Oh, this is a good way of looking at everything. So yes, I'm gonna generate the null and get the results at the same time. Oh God, I forgot to import so many things. I just typed it actually. And then, yeah, so now, there you go. We're computing the results. As you see, it's much faster now. We're going to do this. There you go. Then we have all the results with the correlation and the p-values. You see, most of them are not significant, except for these two I was talking about. Yeah, and I don't know why they're significant, honestly. It's up to someone to investigate the relationship between norepinephrine transporters and functional connectivity, but I don't know. And notice that we can do the same thing with the big brain cortical layers as well, right? Because like, as I was saying, there's actually transformations for the Schaefer atlas to the big brain. So you could just parcellate the big brain data using this big brain version of the Schaefer atlas, and then parcellate your data, and then use this parcellated data from the big brain to do whatever you want using all of these different maps that we have. And so right before finishing, actually, I'm going to just show you averaging, how it works, because it's another very cool toolbox. So you might be like, I actually go on the read the docs just to show you what it looks like. So this is very well detailed. As always, when Ross writes a toolbox, it's always super intuitive, and there's always a very great documentation. What I'm going to use is one simple function to get the data. So I talked about all the different processing steps earlier, but you won't have to handle anything like that, because all you'll need is this function, averaging, that get expression data. And then afterwards, you have all of the processing steps are here as parameters, and you can select which one of the processing steps you want to use. And it's very well detailed, like what these processing steps are. So for example, prob selection, you have to decide like which prob you want. You can either like take an average of all the probs, or you can do a bunch of different things. You decide how to threshold when you so this is like to threshold and remove genes that are not consistently expressed across subjects. You can decide how you threshold this parameter. You decide whether you want to mirror left and right data in the Alan Newman Brain Institute, because in this atlas there's actually 6 bilateral brain, but only 2. No, there's 6 brains, but only 2 that are bilateral. So sometimes if you want right hemisphere data, you might want to mirror the expression that you have on the left or to the right hemisphere. This is obviously not perfect, and so that's why it's actually set to none by default, but it's an option you have. And then you decide how you handle, for example, the missing centroids, as I was explaining. You can either interpolate, and the missing parcel, can either interpolate or match them to like the nearest centroids you have, and then you have so many things right here. So so as I was saying, there's all the steps that we've seen earlier. So, but we've actually selected for you guys like basic default parameters, which should be okay. But like, I think if you end up using this data, I highly suggest you read a bit on the processing steps so that you're actually familiar with what you've done, and you make sure that like it actually works for what you want to do. And it's basically and all you need actually for running this function is 2 things. It's an atlas. So here I'm using again the Schaefer atlas, defined in FS average space, and information about the Schaefer atlas. So this information is basically a Panda dictionary data frame, that stores information about the parcellation you want. So the information in it is like that. So we have the ID of each region. So 1 to 100, as simple as that. And you have the hemisphere of each parcel. So half of them is left, half of them is right in the Schaefer case, and the structure that it belongs to. So all of them are cortex. So the idea is that you simply just want, when you do the centroid matching, you basically want to match left hemisphere centroids and parcels to left hemisphere. You don't want like just swap between hemispheres, and you don't want to assign an expression of a subcortical region to a cortical region afterwards. So that's why you have to specify which cortex each parcel is, which structure each parcel is. In this case, it's just cortex, but sometimes you have parcellation that also incorporate the subcortex. That's when it's going to be useful. So that's all you need. You need the map, the atlas, and the information, and then you run this. It does take a couple of minutes to run because it fetches the data and it downloads it and then pre-process everything. But when you have it, I've stored it already as a CSV file. And then did I forget to? This would have been unfortunate. Expression. Oh yeah, that's because I made a mistake when I write that expression. That's how it's written. So there we go. So I've loaded it, and then we can actually go take a look at what it looks like. So it's as I was saying earlier, it's basically a matrix. So in this case, it's a data frame stored as a CSV file where we have each index, so each brain region, and every single gene. So the size here, we can see in the corner here, it's like 15,000 genes. So it's quite a lot of genes, expression for each brain region. So yeah, 15,000 right down here. So, and then what we can simply do is look at the expression of specific genes. So we can look, for example, SNCA gene, and then we get this parcellated brain map of this gene expression. And notice that in this case, I didn't specify that I wanted to fill in the missing parcel. So like this parcel, for example, there's no data in it. It's not surprising. But if you want, you can decide to use interpolation techniques by specifying it to the function call to get expression data. And yeah, so that's basically how Abidjan works. As simple as that. It's one function call. And yeah, that's pretty much what I had to show you guys today.
Got a quick question.
Yeah.
Do we go hypothesis driven regarding the analysis? So we have like 15,000 genes that we have expressions for them for different brain parcels, and we have a connectivity matrix, for example. And we run analysis, so we do multiple comparisons, but like we do explore and see like which comes out, and then we try to like understand the detail or the background story behind it, or we go like how do we usually do the analysis in this? Because we might find like 100 of genes associated with our like metric of interest?
Yeah, it's a great question. So it's a great question. So it depends, right? So like it says when you do many types of multiple comparison, things like that with genes, when you use genes, oftentimes that's what you end up with, a bunch of correlations. And so it depends how you want to do it. Like if you want, you can end up with just a bunch of genes and then do whatever you want, like you that are significant, I mean, and then research them. But like one of the things that is cool to do that many people did, it's called Go annotations. So when you have a bunch of genes that are significant, I hope, yeah, okay. I was afraid I was gonna miss it, but yeah, so an example of things you can do is you use those Go annotations. So basically when you have a bunch of genes that are significant, you can take this and input your genes that are significant, and they're going to return you annotations of your genes that tell you what they're doing actually in the brain or whatever. So for example, they'll say that it's associated with, so you have terms associated with molecular function, so telling you what they're doing, what they are, so the cellular component they're associated with, and so on, the biological process they're associated with. So you can actually input that, your significant gene. But this is just this is just what I thought of when you asked this question, because this is just one of the examples you could do. But there should be many other things you might want to do for your research.
Okay, thank you.
Show the annotation. So you show that you can fetch your atlas, and then you can fetch, for example, for the Sherpa atlas, you showed that you can fetch the specific version of the Sherpa atlas you want to look at. How do you handle the labels of the annotations in the background then? So do you keep the record of each version, or is there a version independent one for the labels? How you can add labels to the annotations?
So if I understand, you mean like if you have like all the annotations, we only have one MNI 152, but we don't specify which version of it we have. Is that what you're asking? Not sure.
No. So you show the Sherpa atlas annotations, right? That you
Yes.
you still have that open, I think. Yeah.
You mean like here, right? Or
Yeah.
like here?
Yeah,
Depending,
and then
yeah.
and then you show that you have for each of the annotations, you assign that they're on the cortex, but each of the Sherpa annotations actually have labels, right? So where do you store the labels? The terms to name those annotations.
Yeah. It's on the the convention is like in the user guide. Define a persolation. It's unfortunate because it's actually like you have to do it yourself, unless I'm mistaken. But they kind of describe to you how to supply, or maybe, okay, so there are valid label tables already. No, I'm not sure what this is. But what I've done personally, I don't know like if they are available to you, but I've created it myself by looking at this information here. So they specify which structure are available to you, and like how to create basically the information file. So yeah, I've generated it myself, and I've stored it as a CSV file, which I've loaded afterwards.
So do I understand correctly that you don't have the terminologies for the annotations of available atlases already stored as part of your module, but you would have to do this manually afterwards for your current project?
Yes, but yeah, exactly. But this is available. Like, I mean, if you have the Schaefer atlas, you can see which parcels are in which hemisphere. So basically, you'll have to generate it manually. But actually, it's a good point that we maybe should. I mean, maybe they're already available, honestly. But if they're not, I'll definitely work on that so that it's available, so that you guys don't have to generate it yourselves. It's going to be much easier afterwards. But yeah, as far as I'm concerned, I've generated them manually. Yeah.
Yeah, thanks.
Well, thank you very much, everyone on both sides. It was a pleasure. I hope you learned new things or you were inspired by something I said. And thank you for organizing this event. Super.